thanks, everyone. It really is wonderful to join you. And we're going to be talking today about the dermal papilla, and it's really a fascinating subject. And I'd like to review really one very important paper that's come out recently, but I'd like to take you through a story about the dermal papilla over the last 60 years. It really is a fascinating story, and I'm sure one that you know. Many in the group know it quite well, but I'd like to share with you this story and how the dermal papilla really is the control or the boss of the hair follicle. And I don't have any conflicts about anything I'm going to talk about. We're going to come back to this paper by Deng and colleagues in the Journal of Investigative Dermatology in January in just a minute. It taught us that during the course of balding, that the dermal papilla blood vessels die. And you can imagine anytime in human biology that something dies, it's not a good thing. And it's certainly not a good thing that the dermal papilla dies, but it sort of gets us thinking in a whole different way that, we always talk about blood flow. We want to promote blood flow. Sometimes we think it's a little bit exaggerating that, many of the therapies we don't do, many of the therapies that we do aren't just about promoting blood flow to the scalp, but maybe there's more to the blood flow story than we realize. So we'll come back to this in a minute. So androgenetic hair loss is all about the process by which big hairs become thin hairs. We call that miniaturization, as you're well aware. And so when you look up close on the scalp of someone with androgenetic hair loss, you really want to prove to yourself that you're seeing this process by which big hairs become thin hairs. And so here we have a number of thick hairs on the scalp. These are probably 75 or 80 micrometers in size. And then some of them become pretty skinny or thin, 20 micrometers, 40 micrometers. And if you have enough of them, then we call that androgenetic hair loss. And we know androgens are important, especially in males. We know that there's this group of individuals in the Dominican Republic which lack the 5-alpha reductase gene. And these were fundamental studies in the 70s by Dr. Imperato McKinley, showing that, wow, when you lack 5-alpha reductase, you don't get much in the way of hair loss. And you don't get much in the way of prostate enlargement as well. And so this builds on these studies of Dr. Hamilton in the 40s that androgens are important, androgens are important. And so the story is building that, yeah, androgens are pretty important. And then of course the anti-androgens came out, which are hard to refute that they are very valuable in the treatment toolbox for androgenetic hair loss. Of course, we have to, deal with side effects, and they're not perfect, but we know androgens are very relevant to the story. And so here we have the dermal papilla at the bottom of the hair follicle. And it's come to be realized that this is where the androgens are acting. And the story over the last 20 or 30 years has been androgens find their way to the dermal papilla. They bind the androgen receptor. And then the dermal papilla produces these factors that either tell the hair to grow or tell the hair to stop growing. And in the case of androgen binding to the androgen receptor, the message is stop growing. And so the dermal papilla is a key regulator of the hair follicle. There's other regulators that are being explored, including these dermal sheath cells. The dermal papilla doesn't divide, but the dermal sheath cells do divide, and they sometimes under certain circumstances can replace dermal papilla cells. And we know that both of these types of cells are important as we think about regeneration and the ability to make new hairs down the road, and they're both being actively studied. And so when you look at the histology of the scalp, you see this pear-shaped structure with vertical sections through the hair follicle. And this pear-shaped structure is the dermal papilla. It's essentially fibroblasts. And usually in the scalp and in the skin and in many tissues of the body, fibroblasts they're not that interesting. They're there, they produce stuff, but we just see them. But in the hair follicle, they're really the key cells. They're these specialized fibroblasts, and they tell the cells above it, which is called the matrix, what to do. They say, make hair, make these keratinized structures that are going to be 80 micrometers in size. So the dermal papilla is really important. And so let's take a look at this structure at the bottom of the hair, and we'll use this cartoon to look a little closer. The dermal papilla in blue and the matrix in brown, and the hair follicle, the shaft, in orange. So we're making orange hair. This is orange colored hair. And so we think that the dermal papilla and the matrix talk to each other. The dermal papilla seems to do more talking, and the matrix does more listening, but there is some crosstalk that goes on. And in some circumstances, the dermal papilla says, grow, grow, and that makes a hair. But we know that there are a number of situations where it says, stop growing, stop growing. And that's the case in androgenetic hair loss, that the dermal papilla makes proteins such as TGF-beta, DKK1, DICOF1, which tells the hair follicle not to produce a hair shaft, and these are growth inhibitory. It tells the hair follicle to, let's get out of the anagen phase, let's rest now, it's time to rest. And so TGF-beta and DKK1 are really important. And if we can inhibit them, which researchers are looking at, we can get the hair to stay in the growing phase. And so every diagram of the hair follicle shows these blood vessels at the bottom, and they come in and they feed and nourish the dermal papilla. And they're clearly really important. And we're going to take a look at that today. So the dermal papilla is really a very relevant structure. It regulates the development of hair follicles. It talks to the bulge region. It is also a source of stem cells. And we'll continue this evolving story in a minute about the dermal papilla. It really is incredible. And it's a 50-year story in some ways. Of course, it goes back even further, but there were some pretty pioneering studies in about 50 years ago showing that when you take rat whiskers and you take the dermal papilla from rat whiskers, that you can produce new hair follicles if you inject these rat whisker dermal papilla. And these are studies from 1966, 1970. So we're about 55 years ago. And it would be about 30 years until the human studies came out showing, wow, we can do this in humans as well. And Dr. Jahoda, and colleagues, really led the way in some of these studies showing that dermal papilla, dermal sheath cells as well, but dermal papilla injected into the skin can tell the skin to make new hairs. And that's really important information. And we'll take a look at the 1999 study by Reynolds, which Dr. Jahoda is last author. So here's Dr. Jahoda. And I have great respect for Dr. Jahoda, but this is the only picture that I could find. So Dr. Jahoda did a very fascinating study, and it was published in Nature. And if you don't know about it, you should. It's an incredible study. And I don't think we see many of these kind of studies anymore. Dr. Jahoda took his own cells. He took his dermal sheath cells out of his scalp, and he isolated them, and he injected them into the arm of Dr. Reynolds. Dr. Reynolds is Dr. Jahoda's wife. So what was discovered after additional injections is that Dr. Reynolds eventually grew a hair. And Dr. Cristiano, who's also on the paper, said, let's figure out where the DNA comes from. Is the hair female or is it male? Did the dermal sheath cells that went in the arm produce male hair, or did it stimulate Dr. Reynolds' hair follicles to make Dr. Reynolds' hair? And it was female hair that was discovered, and male dermal papilla cells. And so a really important study showing the inductive properties of dermal papilla and the fact that there was no rejection. And hair follicles are really interesting structures because the immune system sometimes lets it go under the radar, and it doesn't necessarily always promote rejection, and we call that immune privilege. So that's really important because this property of hair is really important. In disease states, this immune privilege breaks down and the immune system goes after hair, but in the normal situation where homeostasis exists, the immune system doesn't go after hair. And so the thought at that time was, wow, we can take dermal papilla, put it into scalps, get hair to grow. We have endless supply of cells. And in mice studies and rat studies, that seems to be the case. These dermal papilla cells cheer for months and months and months and months at a time. Let's make hair, let's make hair, let's make hair. But in humans, they cheer for a little while and then they don't cheer anymore. And so we say that the trichogenic capabilities, or the ability to cheer on the skin to make hairs, dwindles quickly. And that's been one of the real challenges in harnessing the power of these dermal papilla cells and dermal sheath cells. And so there's been a lot of interest in understanding the role of the dermal papilla and understanding how exactly does the dermal papilla change in the course of balding. Because if we can better understand how it changes, maybe we can encourage the dermal papilla to stay on board, produce growth promoting factors, and to keep cheering on hairs for longer and longer. And so the key question lately has been, how does the dermal papilla differ in balding scalp compared to non-balding scalp? And so a couple of observations that are relevant. One is that the dermal papilla in balding scalp has higher androgen receptor levels. So the balding dermal papilla is ready to find androgens, to bind, to send these growth inhibitory signals into cells. And these were studies by Hibberts and colleagues, 1998. So we're going to move forward from 1998 to the present day in the Journal of Investigative Dermatology. And so how else do these dermal papilla cells differ in balding scalp? Well, 2008 studies showed that both TGF-beta and here DKK1 is increased in balding scalp, and these send growth inhibitory signals into cells. And in fact, this DKOFT1, DKK1 can cause apoptosis of some of the cells, really shut down the production of hairs. And TGF-beta 1 and DKK1 are key factors in the pathogenesis of balding. It seems that they're not so good. If we could somehow reduce their levels or get rid of them in some manner, maybe we could prevent balding in some way. And there's a lot of intensive research in this area. So in 2008, Bata and colleagues, which includes Dr. Bessem Farjo and Dr. Nilofer Farjo, published a nice study showing that balding dermal papilla are less able to withstand stresses. They're very sensitive to environmental cues. They're sensitive to oxidative stress. They're sensitive to a whole bunch of different stressors. So they're not quite as resilient as we'd like a master controller or boss to be. They kind of dwindle out when stresses are high. Upton and colleagues, same group, showed that balding dermal papilla are less able to withstand oxidative stress. And so the data is accumulating of differences between balding scalp and non-balding scalp. And in 2017, studies showed that these dermal papilla, when stressed, becomes senescent or sleepy. And they're less able to communicate with the matrix. And so the dermal papilla may send signals, but they're not very strong signals. And this mesenchymal keratinocyte epithelial interaction really is not there. And so in order to produce good hairs, you have to have two people talking, a dermal papilla and a matrix. And there's not much talking that starts going on anymore as balding progresses. And a recent study showed that the mitochondria inside the dermal papilla, these powerhouses of cellular metabolism, are altered as well. And so there's dysregulation of these mitochondria. And so as you're thinking about the balding process, there are a number of possibilities you can imagine that's going wrong with the dermal papilla. And probably your favorite pathway and something you're wondering about could be very relevant in the dermal papilla. There's a lot that's not quite going right here with these dermal papilla, and they're not communicating properly. And so we come to this study in the Journal of Investigative Dermatology in January online, showing that in the course of balding, the dermal papilla blood vessels vanish. And that's the term the authors used. Kind of like that term. They vanish. The blood vessels in the rest of the scalp are okay, which makes sense because we don't see necrosis in the scalp when people are balding. So we know the other blood vessels must be fine, but the little blood vessels that feed the dermal papilla are dying. And so this group looked at differences between non-balding scalp and balding scalp, and first looked at genes that are differentially expressed, and they found there's about 1,600 genes that are differentially expressed. There's some 900 genes that are upregulated in the balding scalp and 600 or so genes that are downregulated in the scalp. They showed that the androgen receptor is increased in balding scalp, which is in line with prior data. And they showed that a lot of this androgen receptor is moving into the nucleus. It's accumulating in the nucleus where it can do its job, where it can bind to DNA and affect transcription. And they also showed in balding scalp, there's a number of blood vessel or microvascular abnormalities. And so if we think about it pictorially, this is the normal scalp where we have the hair fiber, the dermal papilla, we have the blood vessels, we have the stem cells in the bulge, we have the sebaceous gland. It seems that in the course of balding that we lose these dermal papilla blood vessels. But the blood vessels in other parts of the scalp are just fine. Something special about the dermal papilla blood vessels. I'll go into this more in a moment, but what the authors showed is that there's an increased production of TGF-beta, TGF-beta 1, and TGF-beta 1 causes these blood vessels that are supposed to be here to apoptose or die. And so TGF-beta is very relevant because it triggers the death or apoptosis of these blood vessels. And it also sends signals for the hair follicle to move from anagen into catagen. It sends signals for the hair to stop growing. Now is not the time to be growing anymore. And so the key findings of this paper were that the androgen receptor is elevated in the balding dermal papilla. There are these microvascular anomalies that exist. They showed that the blood vessels start disappearing at the earliest stages of miniaturization. And so as this hair follicle is going from 80 microns to 70 microns, that's when the blood vessels start disappearing. It's not a late finding, it's an early finding. And we'll come back to the importance of this in a minute, but you can imagine that if blood vessels are disappearing early on, then perhaps that is the stage when we want to intervene in some of our treatments. And maybe there is some pressing time factor here if we're going to save these dermal papilla blood vessels. And again, the rest of the scalp blood vessels are just fine. So it's not a general issue of blood vessels. This paper is very nice because there's about 25 elaborate experiments in this paper. And so the author showed that angiogenesis is really important for hair cycling. And as hairs go into the anagen phase, there's an upregulation of vascular endothelial growth factor as part of normal hair cycling. And they showed that when you inhibit VEGF, the vascular endothelial growth factor receptor, you block hair cycling. And so it's very clear that angiogenesis is really important to allow hairs to cycle. It's very much a part of the pathway. TGF-beta was increased in the balding scalp when compared to the non-balding scalp. And so it does appear that androgens bind to the androgen receptor and are able to trigger the apoptosis of blood vessels and trigger the quiescence of hair follicles. And so the authors do propose in their final statements of the paper in their concluding section that, maybe we should be treating androgenetic alopecia earlier than later on account of saving these blood vessels. And so I think this is a really nice paper. It really highlights not only some of the subcellular structures that the literature has looked at, mitochondria, oxidative stress, metabolism issues, but it looks at some of these blood vessel issues as well. And when you look at some of the treatments that we currently use, one needs to ask themselves, how are these treatments affecting blood vessels? We'd like to save these blood vessels. I wonder if any of these treatments help blood vessels. And certainly minoxidil can activate angiogenesis type signaling or VEGF signaling. And so it can promote a more blood flow friendly state. We use anti-androgens, of course, in the treatment of androgenetic hair loss. And by blocking androgens, theoretically, we would expect that we are shutting down the production of TGF-beta, and we are theoretically stopping the death of blood vessel cells. And so many of these treatments that we're currently using probably have a very important role in how they affect blood vessels in the dermal papilla. A lot of the treatments that are currently looked at, including affecting the Wnt pathway, blocking DKK1, are certainly very relevant, and other anti-androgens that are on their way. And one certainly wonders about this TGF-beta pathway as well. And TGF-beta has been a little trickier. TGF-beta does so many things in the skin. Under certain circumstances, it promotes proliferation. In other situations, it inhibits proliferation of cells. So it's a very interesting protein. And there had been TGF-beta inhibitors that came to market for other indications years ago, were studied and haven't found their way, but there has been an interest from time to time in what TGF-beta does. Certainly in this dermal papilla microenvironment, it would be wonderful to shut down TGF-beta, but that really hasn't become a mainstay research area. And so this is the story of 55 years of thinking about the dermal papilla, right from the very early days, taking rat whiskers and showing, wow, they induce follicles formation, to human studies showing that the dermal papilla can induce hair follicles. And so there really is a tremendous interest in understanding the dermal papilla, because it's very clear that under the right circumstances, the dermal papilla's potential is vast. And so understanding the differences between balding scalp and non-balding scalp are going to continue to be really helpful as we understand how how to get this dermal papilla in the happiest state possible, where it can send growth promoting signals to the matrix rather than growth inhibitory signals. And so that's where I will stop. I want to thank the ITC again for this kind invitation, and I'm happy to discuss anything about hair that you'd like. Thanks again.
Thank you so much, Jeff. That was a fantastic talk. If you just want to unshare your screen for a second. Dr. Jeff Donovan's podcast. Now, it's really difficult to get a podcast together or a blog together on a weekly basis. Now, Dr. Donovan does it fantastically. Now, this is a lesson each week. I would highly recommend that you go onto your designated podcast find this, and it's a lesson each week. So thank you for that.